Good morning, everyone. Thank you for joining us. I'd like to reward those of you who have sat through some technical talks this morning with a few familiar faces. So my role is, I'm a salt marsh ecologist on the project, and what I was tasked to do was to try and relate these carbon fluxes that seem abstract and are dynamic and have complicated equations to some of the simple community players that are present in a salt marsh. And so I'm talking this morning to you about some of the work we've been doing trying to relate these processes to the plant communities in marshes. So I'd like to begin with just a brief overview of what we know about salt marsh zonation as it pertains to these gas fluxes, and then I'll show our data that has been comparing them between major plant-defined zones at a reference site, Sage Lot Pond in Wacoit Bay, and then I'd like to hopefully open up discussion by presenting a few example applications for how we might use this type of data. So here's those familiar faces. How many of you can name one of the species shown in this diagram? Anybody chased a fiddler crab through the marsh? That's what I thought. So this was my starting point, really, when we began this project, that I was aware that there were interesting patterns in the plant communities and the animal communities in the salt marsh that are driven by an interplay between physical parameters and biological interactions. And these are some of the environmental factors that vary as one moves from the smooth cordgrass, low marsh zone up to high marsh communities where there's greater plant diversity. First of all, salinity increases when there's higher rates of evaporation and less tidal inundation. And so these are just some average numbers in terms of salinity. Shown here between the high and low marsh zone in terms of moisture being higher in the low marsh zone, redox potential, which is a measure of how much oxygen or energy is available to decompose organic matter, being lower in the low marsh zone, and sulfide concentrations being much higher in the low marsh zone. And we have known for some time that that influences plant morphology and that these plants have unique adaptations, such as parenchyma, shown here, which are spaces through which gas can transport through the plant tissues. It's useful to plants because it allows oxygen to penetrate down into what would otherwise be waterlogged anoxic soils, enabling them to obtain energy through respiration and to withstand some of these very high sulfide concentrations. And so those types of properties that facilitate oxygen exchange also, of course, impact greenhouse gas transfer. This is just a couple of pie charts showing what the community composition of the plants looked like between high and low marsh zones. Our low marsh is dominated by the smooth cordgrass Spartina alternate flora. And the high marsh community is a mixture, in this case, of Disticus spicata, which is the light brown color on the top pie chart, and Juncus gerardi, which is the darker brown color. And these are some illustrations of what those plants look like. I show this because the morphology is interesting in terms of ecology. The way that some of the plants like Disticus can extend runners affects their ability to colonize open spaces, but also the morphology, and particularly the plant, the below ground biomass morphology influences carbon. Those roots are exuding carbon as the plants photosynthesize, and that root material is also potentially contributing to these long-term stocks of carbon. And so we know that the plants, because of those differences in the physical environment with zones, they have physiological responses, and I've shown the parenchyma as one adaptation for oxygen transport. And these are just some data that are illustrating that oxygen transport occurs through roots with very high rates of oxygen being exchanged by some of the low marsh plants where you would predict it would be needed, and that the plants are also responding to the physical stress. In this case, this is just a measure of leaf acerbate peroxidase, which is one enzyme that indicates stress response to salinity and low water, and that was actually higher in some of the high marsh plants. So besides the basic zonation pattern of monospecific zones as one moves from low to high spots with elevation, there are patches within the marsh landscape. They're not quite as simple as one might think, and some of those patches are influenced by waterlogging, for example. And this is a nice experiment of many that I could have brought forth to illustrate that waterlogging, for example, can create new bare spaces that allow some less competitive species than the grasses to come in. And so this is an example of one forb triglocan that is thought to be decreasing as some of the ditches have been built into salt marshes, cutting back on the extent of waterlogging. So those dynamics, as well as interactions that plants have with microbial communities, are going to be influencing greenhouse gas fluxes. And this is a cartoon I've constructed just to illustrate that there are many microbial communities involved in the cycling of nitrogen, for example, around plant roots. That are interacting to affect emissions of one greenhouse gas, nitrous oxide, that we'll be talking about tomorrow. And what I'm looking at today are some of the microbial plant interactions that may have been influencing methanogenesis. And all of these activities are different ways of decomposing organic matter that can result in release of CO2. So the plants are interacting with these microbes below ground, and then they're acting as conduits for the gas products, actively transporting them up to the atmosphere. And finally, we know that some of the zonation patterns that I've just described are changing in response to human impacts, development of land, changes in nitrogen loads, and introduction of invasive species. Whoops, I just turned that off, that was fun. And one of the things besides waterlogging that is influencing the change in zonation is actually a longer term global phenomenon with sea level rise. This is of interest to us in this region because we're in the hot spot for relative sea level rise rates, these being particularly higher than average due to a combination of physical oceanographic conditions and being exacerbated by very low sediment supplies to build up the marsh. So this, of course, begs the question of what is the impact on blue carbon. As we start to see what look like signs of sea level impacts, that the waterlogging that was once part of the just basic ecology of the system is becoming more pronounced, more prolonged, and instead of leading to new vegetation is leading to bare vegetation or standing water. So again, my role is to ask whether plants are simple proxies for our blue carbon efforts. It's terribly expensive to measure gas fluxes, but wouldn't it be lovely if we could characterize the plant communities instead and have some idea of how much carbon is being exchanged in the system? And to ask the question for methodology purposes that you'll hear more about this afternoon of whether stratification can improve accuracy. That means, can we break the system down into different components, and if you do a different way of accounting for each of those components, do we get a more accurate answer? And finally, from a scientific standpoint, we're of course interested in this in order to get at some mechanisms. So you've already seen the study site. My species comparison was focused only in sage lot because that's our pristine reference, which had very little nitrogen inputs. And there we have been studying different plant-defined zones since 2012, including what I'm calling just the high and low marsh communities, as well as some of the unvegetated ponds and the upland Phragmites borders. You've seen the boardwalk already. I'll just show you some pictures here of the different types of ponds that we've been studying. I looked at ones that varied in terms of depth and therefore potentially age. We used a different custom chamber designed by my soon-to-graduate PhD student, Rose Martin, who has been focusing in Phragmites, and she'll be presenting a poster this afternoon. And we used floating chambers to quantify some of the emissions from the ponds. There were many data collected, and I'm showing you only a subset of these regarding plant properties, soil properties, and climatic properties. So this is a snapshot of the zone comparison just between low and high marsh. The darker colors are indicating CO2 fluxes that were measured in the low marsh community on a series of dates. And the lighter colored bar next to them are the fluxes from the high marsh zone right next to them. And the take-home point here is that the low marsh definitely had higher rates of CO2 uptake. And these are instantaneous fluxes. Remember, this is not long-term burial, but this is what you go out and measure as a net exchange at any given moment on average between multiple spots within these zones. And then methane, in contrast, did not have a significant difference between the zones. There's something else driving that variability. And clearly, there's a strong temporal difference with methane and high variability. That's the challenge of working with that particular gas. In general, these numbers are much lower than you see in other systems, and that they are lower than the CO2 exchanges, and so that's good news for blue carbon efforts for salt marshes if these methane values are low, even if they're tricky to pinpoint. This is just a snapshot of some of the temporal variation that we've seen. That what you're seeing in the blue bars are CO2 uptake rates that we've observed throughout a year from June to December. This is a winter period here in the middle. These are some of those bi-monthly measurements that were taken during daytime, and then the green bars are the corresponding values at night. And so what this is showing is that there is respiration at night. That's not surprising. It's interesting to look at the magnitude of it because that allows us to confirm that if we want to do a budget for carbon accounting, we do need to consider the nighttime respiration rates. It's not trivial compared to the daytime uptake rates. And then methane is also, again, not quite cooperating in being predictable based on day or night patterns, and that's the basic take-home point message here, though clearly there's some temporal variation. So looking at the potential connection to sea level rise and water logging, I'm comparing here fluxes, CO2 fluxes between ponds that have permanent standing water, high marsh native vegetation, and invasive Phragmites vegetation. And what we're seeing is that, first of all, there's not much going on with the ponds. There are some small emissions. The fact that there are emissions is interesting because compared to the native marsh, there would be uptake of CO2. But the very big, perhaps shouldn't have been as surprising as it was, finding was that Phragmites, this invasive species, actually has much higher CO2 uptake rates than the native vegetation. And when we look at the methane data, it's a bit less predictable. Methane continues to be variable. It is highest with the Phragmites, but as Rose can tell you in more detail with her poster, it was relatively small compared to CO2. So if I could try to just give a relative magnitude comparison of these different plant-defined zones, it would look like this, with the biggest arrows, downward arrows indicating CO2 uptake rates, exchanges of CO2 being drawn down from the atmosphere into the soils. These numbers are after some correction for this relatively small methane fluxes that are observed. And what we would say is that the hotspot for CO2 uptake would be the low marsh Spartina alterniflora zone and the invasive Phragmites zone. And that potentially some of these ponds are actually sources of CO2 rather than sinks. I did some ballpark estimates taking into account our light and dark data and our temporal data over the cycle of a year, and estimated this number for an annual CO2 uptake rate. And I've just aligned it here with the range of CO2 sequestration that is observed over decades. So my data are based on instantaneous measurements extrapolated to a year. And this number will be presented by Dr. Ganea tomorrow morning, which is based on longer term dating. But they're in the same order of magnitude, and probably the difference there is certainly influenced by some of the lateral fluxes that are going on, if not other properties as well. So to summarize our plant simple proxies, unfortunately, I think the answer is no, but they certainly can be helpful in improving accuracy of our carbon accounting, because we did see that some zones have much more much higher fluxes than others. There is more work to do, frankly, to understand the mechanistic causes behind these, but it looks clear that invasion may actually maintain or increase CO2 uptake, while sea level rise, if it causes waterlogging that we've been observing, would tend to decrease that CO2 uptake. So finally, I hope this will be of use and interest to you. I've just tried to brainstorm some ways that we might take these data and use them for coastal management. And so one way is that we can use it for scaling up. I imagine that many of us have access to habitat maps like the one shown here, produced by GIS, that are showing the representation of different plant-defined zones. And so if we know characteristic emissions or flux rates for each zone, we can ask the question of what are the hotspots. And what I've done here is just to calculate an estimate of the overall carbon uptake rate in terms of grams of carbon per hour from each of these zones. And the light green in both the pie chart and the map is the alterniflora or low marsh habitat. So this is for Sagelot Pond. And if you're working at a series of sites, then you could do a site comparison, such as this one. What I'm showing you here is Sagelot on the left, and then Hamblin Pond, one of our higher nitrogen loading sites, on the right. In both cases, the low marsh really is dominating the overall rate of carbon exchange. However, Phragmites significantly differs in terms of its role. That's a function of both the area that they're covering and then the area-specific carbon rate. So this might be useful to apply not just for site comparisons, but in evaluating what changes you think may occur with restoration before and after restoration. If, for example, you can predict how plant habitats will be changing. And then another way we can use these data, for those who don't want to haul around a 200-pound analyzer in the field, is through modeling. You can take data on things like plant biomass and see how well you can do to describe the CO2 fluxes in a model. And Dr. Abdul Aziz will be taking you through that this afternoon and explaining how that is going to be physically made available. But I highlight this result, which is showing you that below ground biomass, which is highlighted in red here, had a significant relationship to CO2 fluxes in one of his preliminary models, playing a stronger role than above ground biomass or stem density. And we're doing similar models that are also incorporating basic soil parameters. This is a nonlinear model, and this is just showing you a plot of the observed data versus the model data, and it's fitting really surprisingly nicely. So I have many future directions for research, but I think that our keynote speaker outlined and exceeded them, and so I'll just open up at this point for any questions or comments. Thank you.
Yep, sorry. So in some of the pools up in Maine, we see submerged aquatic vegetation, so plants like rupia or other algae, for example. So I'm wondering if in any of your pool comparisons, you looked at pools that had plants versus not, and how you think that might change your emissions.
Yeah, so many of the ponds that we looked at had microalgae, but I would not say they had any substantial macroalgal mat growth, and it's definitely not seagrasses such as rupia. And I would predict that it would increase the CO2 uptake rates because there's photosynthesis happening there. But yes, it certainly is a next direction to go, and I don't envy the seagrass community for having to deal with subtidal chamber deployments. But I'm happy to help if you'd like.
Very nice presentation. And I was happy to see those microbes in your model. As a grandmother and an educator, can you please help us to do something like this in other countries? We need that information. But in the United States, can we, the grandmothers, understand what's the interaction with, let's say, chemicals that we use to actually kill invasive species? Let's say, for instance, glyphosate. We use it for, the Roundup that we use for keeping our lungs beautiful. So, but glyphosate is an antibiotic. So how is that bacteria changing over time? I just would like to see some of that data incorporated there. And for the grandmothers of Massachusetts, we are willing to partner with NOAA to pay for instruments, simple instruments that will gather this data for the 3 speakers that just presented. We mean business.
Thank you, Dr. Warren, for your suggestion. I know that there's a need to branch out beyond the New England region with much of this research, and certainly some of the reviews have indicated mangrove ecosystems are quite important, and we know that there's a lack of data there compared to salt marshes.
Thanks, Serena. I really enjoyed your talk. This is Tom. So I have a question. I know that you showed a really high carbon fixation rate at both ends of the spectrum, whether you're at the low marsh or the high marsh. And I was just wondering if that can account for the differences in DIC export that Kevin was presenting, or are these 2 completely different sites?
They're not 2 completely different sites. That's a good thing, I think. And I don't know how exactly that can account for the differences. We definitely are poised for that conversation if we can begin it. As Kevin mentioned, he presented some of his data for the first time to us this morning. And I've been wondering about how we can connect that. But I think that one approach we might take is doing a weighted average, knowing the representation of the different plant zones across the marsh landscape, and knowing the drainage area for the lateral flux component that he is using. We can ask the question of whether Phragmites has any potential influence on that drainage area, and if not, then I guess that simplifies the answer.